Russell, that was so far from being dull. You have no idea. That was a fascinating presentation. I know everybody in the audience, myself included, was absolutely enthralled and applauds you on the great work that you guys are doing with the nanoparticle template. Very, very fascinating. Our next speaker, continuing on the theme of medical reasons to develop nanomedicine, is Rutledge Ellis-Behnke. Dr. Behnke graduated from Harvard Business School's International Senior Managers Program, as well as receiving earlier his PhD from MIT. Dr. Ellis-Behnke was the co-founder of the first online commerce company in computer memory, as well as senior vice president of a public testing and consulting company. Currently, Rutledge is working in a really fascinating area. He teaches at Hong Kong University Medical School and at MIT, a lot of travel there. His work uses nanobiotechnology to reconnect disconnected parts of the brain to the spinal cord. A great example of this technology and of the medical reason to develop it. Please join me in welcoming Dr. Ellis-Behnke.
I'll try not to step on that. I'd like to thank the organizers for inviting me on this. Now, I'm going to go through a lot of slides, and rather than go into a lot of detail on all of the data, you can read a lot of this in the papers. What I wanted to do was I wanted to go through, one, the motivation behind what I do, so you know that I'm a neuroscientist. And if you ask me really hard questions about the chemistry, I can probably answer about 80% of it only. So be kind. I always put this in. I've been very fortunate that we've gotten quite a few patents out of some of the work, and some of it's already been licensed, and Clear Nano Solutions is just changed its name to Arch Therapeutics or something like that. So at least that's what I was told. As you know, MIT has a great policy for patenting things. So let me start here. I translated this. It says central nervous system regeneration in hieroglyphics. And I did this. The inspiration was the Edwin Smith papyrus from 3000 BC, which was one of the oldest references I used in my thesis. Now, I just have to correct one thing. I finished Harvard Business School, and then I went back to MIT to get my PhD. I only had a PhD for about 5 years. So what you're seeing is a lot of work for my PhD and then follow-on work. And I'm going to come back to this as the last slide because the Egyptians were going through the exact same thing we're going through right now, only at a different scale, and I'll explain that later. Here's a self-assembled system that I work in, the central nervous system, which is the brain and spinal cord, and I teach anatomy and intro to anatomy to people, doctors and medical students, as well as people at MIT. And my whole goal is to reconnect disconnected parts of the brain to restore quality of life. And I don't want to get into that. If you want to ask about it later, I'm happy to talk about it. But that's a different answer for every person depending on what kind of injury they have. I also throw some socioeconomic information in here and a little bit of geology or geography as well. So in the US, there's about 12,000 spinal cord injuries a year, 250,000 people living with it. Severe traumatic brain injury, there's about 1.4 million people that suffer a TBI every year. 100,000 of those every year are severely traumatically injured. There's 5 million people living with severe traumatic brain injury. And severe stroke, there's about 5.7 million people that are living with severe stroke. Now, even though spinal cord injury is a very serious injury, most of those people before the injury are the same mentally as after the injury, where these other two groups are not. And to contrast that to China, 42 million people are living with a disability from traumatic brain injury. 500,000 people die every year. And there's, just to give you an idea, there's only 90,000 worldwide spinal cord injured people every year. 90,000 versus there's 42 million just in one country. Okay, the presentation. I am pretty simplistic. So when you think about this, I try to look at it from how do I approach the problem. So I call it the 4 Ps of regeneration, call it my business background, but to make it simple. You have to think about the brain and nervous system like plants in a nursery. If you cut an oak tree off that's really old, the likelihood of it dying is pretty high. But in order for it to regrow, it has to regrow from that stump. So if you think about that, and then you think about the central nervous system, those two, that analogy I think is very apropos because it has to regenerate from where it was injured. And there are some instances for reanastomosis of injured axons very, very quickly, but they degenerate very quick. So the first thing is you need to keep it alive after it's injured. The second thing is you want to create a permissive environment for growth. If you don't have a permissive environment, nothing's going to grow through it. You want to then promote regeneration of the preserved cells through the permissive environment and then increase plasticity. So this is just a thing. So time is along the bottom. The first one is preserve, permit, promote, and then plasticity. And what you'll notice is at the top here, you want to keep the cells alive and then you want to keep them alive while they're regrowing. You then want to create a permissive environment of the scar and the permissive tissue where it has to reconnect to. You then want to promote growth, and you'll notice that this is about 24 hours later, 24 to 48 hours later, after you have created a permissive environment. And then plasticity you want to increase during reconnection, and in some cases you want to stop the immediate post-injury plasticity right after the injury, because in most cases you have to undo it in order to get functional return. So there's no magic bullet. I'm going to show you one approach. I think the stuff that Tad presented earlier today is very intriguing, and I think it also has a lot of application in a lot of the areas that we're going to be talking about. So this is just an example of some of the things that we've done to go ahead and approach this. Transfections to preserve and also to permit, promote, and plasticity. And these are transient transfections that only last between 2 and 4 weeks. You don't want them stable. Just like early development, you don't have axons growing continually throughout your life. They grow, they connect, and then they stop elongating. They go to arborization or plasticity. Scar reduction to create a permissive environment. Bridging methods, you can do anything from self-assembling peptides, which I'm going to talk about. We've also done sciatic nerve transplants as well. And then training effects for plasticity, basically rehabilitation. You guys have seen this before. I'm not going to go through it. Everything I'm going to talk about today is building structure with molecules, all of it. So keep that in mind. Don't think about this as a promotion of growth. Just think about the creation of a permissive environment and building a structure. And the structure is remodelable depending on the local charge. So this is an arginine, alanine, aspartate alanine. It's 16 amino acids put together in the same way. It's a RADA repeat times 4. You put it with any ionic solution, it will actually go ahead and form beta sheet. And I was really concerned about this when we first put it into an animal because I was thinking self-assembly of a small protein-like structure, prions. That was the first thought I had. So, and I'll come back to that. I put both of these up here because this is one, here's another. And if you look at the molecular structure here, this has a much higher modulus than this one. And that becomes very important when you see later in the talk. So building structure with molecules, it looks exactly like water. It flows like water, 1%. It'll flow into an irregular area and then self-assemble. These are AFMs, 100 nanometers are small fibers. And these are different concentrations, 1%, 4%. You can see that the amount of flow of this is much higher at a lower concentration than a higher concentration. And this is a video. He has to punch this. So basically what I tried to do is I just tried to go ahead and show how these things would assemble. Now, this is a molecular model. This is just one part of it. But basically, the assembly is perpendicular to the direction of the fiber. So it's assembling across the fiber, and the fiber is going out of the board, basically. These are SEMs. Remember, these are highly hydrated. This is 1 micron, 0.5 microns. So it looks like hair in the drain. I haven't seen that in quite a few years. So to pull on that point earlier, if I was 20, I would look exactly the same because I lost my hair around 18 or 19. So So probably 18 would be better. So I'm going to run through this pretty quickly. This is very simple. It's published, you can look at it, but it's 2 experiments. The only thing we're trying to do is see if we can create a permissive environment. The first thing we did was we used young animals, P0, and then we used adult animals. Here's the brain, superior colliculus. For all of you who don't know neuroanatomy, if you see something out of the corner of your eye, you turn, that is mediated by the superior colliculus. Rodents use the superior colliculus an awful lot for vision. So basically what we're going to do is we're going to make a cut, we're going to put the material in, and you can see here we already are starting to get recapitulation of the tissue in the first 24 hours. This is a saline control. So this is the first time we'd ever seen the re-healing or re-knitting of the tissue in the middle of the superior colliculus. 1 month later, again, these are very young animals, 2 days after they're born, so the axons are able to regrow if you can create a permissive environment. Here are 2 controls. Here are 2 right here that have completely healed, and so we see healing. Do we see axons going across? Again, a control. This is a tracer we put in the eye that's fluorescent, and it does not go transsynaptically. Even though there's a bridge here, nothing grows across. And so these are direct projections from the eye directly through the center of this cut and into the superior colliculus here. And I'll blow this up, and you can see these are all axon and axon terminals that have been labeled from the eye. So they've grown back through the center of that cut. So we're not seeing the normal scar response in the brain. So what? Young animals. So we did it in adult animals. You cut the brachium of the superior colliculus. The eyes are out here. We remove the cortex. This is the optic pathway into the superior colliculus. You cut it, you put the material in, and you can see here we have complete healing and regeneration through it. A little bit easier to see. The one thing here, these yellow dots, everyone thinks they're macrophages. They are not. They're actually red blood cells. So we're actually modulating the immune response in that local area at the nanoscale by blocking the immune response, so we're not seeing the red blood cells taken out of the local area, which is quite interesting. I can't go into that yet because it's not published, but it's a fascinating, completely new line of work. So this is a superior colliculus is about 75% of normal reinnervation, and we found with other work that we've done, if we had 42% of local innervation of one small area, that was enough to go ahead and actually drive complete regeneration and return of functional vision. So what's a blind animal look like? It's only cut on one side. You wave this thing, you do it 20 times every other day for 6 months, and you analyze every frame of the videotape. So if you could push that one more time, just back up. Let me try one more. There we go. Just so you see here, you're waving it here, the animal doesn't see it over here. It's a little sunflower seed, they put it in their pouch, they'll do this over and over again. It takes a long time to do this kind of work. This is an animal, second day of testing, 6 weeks after the treatment. We actually see return of function already at 6 weeks. And we found that if you can control when you train the animal, if you train them more during the reconnection phase, you actually get better rehabilitation than if you don't, which comes to some other work that we've been doing. I'll talk about that. I had to throw one graph in because I'm from MIT. Here is an EM. So this is the material. This is the central nervous system. This is 0.2 microns. Here's an axon. Here's another axon. Here's the material. It looks exactly like extracellular matrix. Within 3 to 6 weeks, it's completely gone, comes out in the urine, most of it, or it gets broken down and used as building blocks. This is unpublished work. Chondroitinase ABC, think of it as a lawnmower for the scar. So basically, same paradigm. We take and we put the material in with ABC chondroitinase, and what we see is, this is at 12 weeks, we actually see a higher axon density in the superior colliculus with both treatments as opposed to just a self-assembling peptide. And what we see is we see a much higher innervation density, and this is just a blow-up of that, and these are axon terminals and axons in that area. And I won't show that. Let me go to the next one. Come on. There we go. And what I want you to do is take a look at this animal, because the speed of this animal is much faster. Second day of testing, 6 weeks later. The interesting thing is, what we found with these guys is the material seems to be holding the enzyme. If you put the enzyme in, it's gone within a few hours because it gets broken down. If you put the enzyme in the material at the same time, think of a vase in packing peanuts. It actually holds the enzyme there. It doesn't break the enzyme down, and as the material breaks away and goes away, it continually releases the enzyme over a long period of time. And it's completely viable at room temperature for 9 weeks. So then, how can we go ahead and look at regeneration while we're doing this in live animals? So we have a 7T magnet. Can we see a contrast enhancement agent? Yes. So this experiment was the day one we inject the eye and we scan, then we cut, inject the eye and we scan, we reoperate. So this is a chronic case, 21 days, 7 days after the second injection. Day 66, we then look. Here's the eye. Here is the intact animal. Here's the spirocliculus. You can see a little bit here. This is the second scan after the surgery. You can see a complete disconnection. This part right here is connected to this eye. And then you inject again at 6 weeks. And what you see here is, you see, well, that doesn't look really that good. There's not a lot of reconnection. It's about 15% of the total retina. What we found was that manganese chloride, which is completely safe for humans, when you inject it into animals, it actually gives them cataracts and kills the retina. So we had 80% of the retina dead, but 15% of the total retina reconnected. So we had 5% that wasn't reconnected of the almost completely dead retinas. And this is after one injection, 48 hours. This is after 6 weeks. And these are eyes from an animal, and you can see how much it shrank. For glaucoma, can you look at differential diffusion rates? In glaucoma is high pressure in the eye, and what you see is, you see here in the normal eye, you see this diffusing pretty far. Here you see a constriction rate here brought on by the overinflation of the eye. Using diffuser tensor imaging to go ahead and look at the breakdown. Here's an optic nerve. Here's an intact optic nerve. You can start to see that the diffuser tensor image signal is starting to degrade. So that means that the axons and the myelin are breaking down very, very quickly here. And you can use this as a diagnostic tool to diagnose glaucoma and damage very, very quickly. This is within 7 days. The goal is to be able to go ahead and take an image of someone and figure out exactly what you need to do when in order to get reconnection at the specific times, knowing that every one of the neurons is growing at a different rate and will reconnect at a different rate. So you want to do the right thing at the right time, knowing that's going to be different for every type of neuron. Growth and survival of stem cells, I won't go through that. This is spinal cord. You make a hemisection, you put material in it, and we saw healing of the spinal cord, saying nothing about regeneration. This is interesting. This is a pH issue. So you take neural stem cells, or you put precursor cells in, green fluorescent protein expressing cells in a normal rat. 1 week, 4 weeks, you start to see holes. 8 weeks, you see big holes. We couldn't figure out what this was. And a neurosurgeon did this and basically was perfectly clean, dried everything. So this was a pH effect. As soon as we addressed the pH, this is 2 weeks, this is 8 weeks, and basically what we see is we see almost 100% survival of the cells, and they migrated up to 5 centimeters along the spinal cord. Again, this is just an example of neurofilament right here in red. This is in the center of the cut, so we see cells migrating out, cells migrating in, and these green cells have migrated up to 5 centimeters along the spinal cord. Again, here is an immune response if you don't buffer it. This is, if you buffer it, you get no immune response whatsoever. This is ED1 staining to show any immunogenicity. That's all published. So as we were injecting this into the brain, all the bleeding stopped. And I thought, oh boy, this stuff is so toxic, they're all dead. So you can stop bleeding a lot of different ways. If you make a cut in the femoral artery, here's a sciatic nerve, femoral artery, you make a cut, it bleeds all over the place. You can tell a student when they've done it the first time because they come out and they look like an axe murderer. And you put it on and you can see that the sciatic nerve here is now visible, and you can actually see through this and actually see where the hole is and where the cut was, and the bleeding has stopped. But I'm going to show you a video of that. So here's a liver. I'm going to make a cut in the liver. Let's see if it'll work. Okay. This is 1%. This is something you would never do. You never take a scalpel and make a cut in the liver. You always use some type of cautery device, you use a laser or something like that, which causes huge immune issues because you're causing a burn. Now, these are all real-time videos. I haven't taken anything out or added anything to it. And what you see here is this is a pumping bleeder. I did this, and you move it up so you can see a little bit better. And I'm just going to take and express the material between the two halves. And that's how fast it stops bleeding. The bleeding's completely stopped. You can sew that animal up, and it will go ahead and heal. It'll break down within 3 to 6 weeks or 8 weeks, depending on the concentration. And we've gone to other types of ways to do this, and this comes back to something we saw earlier today. The modulus of the material you put in. If you have something of the wrong modulus, it goes ahead and shears apart. That's in one of my papers. If the modulus is too stiff, it pulls apart. It has to match the tissue, otherwise it won't work properly. I won't go through this one because it's a little bit long, but it's just a bigger bleeder. This is a red blood cell. Here's the material. Here's a hepatocyte. Hepatocyte. Notice the red blood cell is not lysed. Here is a 50 nanometer scale. Here's part of that red blood cell, and this is the material. It does not interact with the red blood cell at all in any detrimental way from what we've seen. This is what a red blood cell looks like drawn to scale of that. It's a physical barrier, which is what we understand so far. It is not acting on any of the clotting cascades, and it's completely compatible. We've had it in over 1,000 animals. And this is something we came up with. The other one's really impressive, but you make a 4 millimeter punch biopsy and you fill it. 6 weeks later, you can see it's completely healed. This is a H&E stain, 48 hours. These are all to scale. This is 7 days, so it goes from here to here in about 5 days. And then 6 weeks, you can see it's almost completely healed. What we see is this material is actually opening up and letting cells migrate in. So if you think about early development, a scaffold that will assemble, and then when you get in close proximity, it actually disassembles and then reassembles again. So it's actually remodeling based on the local environment. And I'm almost done. We've done this in rats and hamsters and mice and all sorts of different animals. The final thing. So you think, okay, you can actually see through it after it stops bleeding. Can you use it prophylactically? Now, this is not published yet. So, and we've done this in a whole host of different things. And basically, what I hope this will show, is this going to work? Yes? No? No? Okay, well, let me just bounce through the rest of this, and at the end, you can just go ahead and bring it up in Windows Media Player. It usually plays that way. Basically, what this would show is you spread the material over, open hole in the skull. It remains a liquid except for the very local area next to the tissue. And as you're operating, it stays completely clear, and it flows in and stops the bleeding as you're operating. And I'll show you that in just a second. Size really matters. I'm going to bounce through these. These are all provided. You guys can take a look at these. These are the same issues you're going to hear over and over again over the next few days. These are areas that we've worked in, the yellow. These all have a component of nanotechnology in them already. And let me so back to this. The reason I put this up here is the Egyptians were building structures bigger than they have ever built before with mathematics they didn't understand and materials they didn't understand. And I think we're at the same place, only we're working in the opposite direction. And we're working with physics that's not very well defined. I think there's going to be more controversy, I think, over the next day and a half about that. But I think this is a big issue. And we're right at the edge of trying to figure out why some things can work and some things don't. Peptides, there's 100 of these sequences that we've used. There's 75 new sequences that we came up with. And there's probably going to be about 400 by the time we're done with all of this that don't have any other vasoactive properties or any other detrimental issues. And they will make very unique structures. So Acknowledgements, a few people I work with at University of Hong Kong Medical School, MIT, and so I spend part of my time here at MIT, and I spend part of my time here at the Medical University in Hong Kong, and I teach anatomy and a whole host of other things. But let's see if we can we play that, just bounce out of this, and you can bounce out of this, and we'll just go to the video. Oh, it should say crystal clear surgery, brain surgery, removal of dura. Oh, here we go. Okay, good. So here's the skull, and you can't even see the bleeding. We thought that this would assemble completely all the way, the entire thickness. But you can see there was a little bleeding here, and the bleeding goes a little while, and then it eventually stops. Okay? Now, we thought this would be a complete gel. What I want you to do is I want you to take a look at the forceps when I come in, and keep an eye on the tips of the forceps as well as the liquid going up on the forceps. I'm going to remove the dura of the brain of this animal, and it's usually very, very bloody because there's huge capillary bleeding. But what you'll see is, after I do a little Blair Witch project here and move it around so you can see a little better, and again, these are real time, so I apologize, my videography is not the greatest. So you see how this is wicking up onto the forceps, and look at the tip. So what happens is you pull the dura back a little bit, and it bleeds, the liquid flows in and it stops. A little more, bleeds, liquid flows in and it stops. But one of the things you notice is the area is completely clear. So we sort of I call this crystal clear surgery, because you can operate through this prophylactically, and it actually flows in and stops bleeding as you're operating, and the material is completely clear. So you can reduce the surgical time by as much as 50% by being able to go ahead and operate through this, because 50% of the time, especially in neurosurgery, the huge capillary issues. The capillary bleeding is the biggest problem. So, and with that, let me just see. There was one slide, no, I didn't put it in, which basically shows the same paradigm of doing a 4mm punch in liver, and we put carbon nanotubes in it, and we ended up getting fibroids growing in the liver. So not all nanomaterials are good or bad. Each one is unique. And with that, I'll say thank you very much.